The topic of this screencast is the digestive physiology of the small intestine. This screencast was designed to help you achieve the following objectives. Explain the following processes that occur in the small intestine. Digestion and absorption of carbohydrate, digestion and absorption of protein, digestion and absorption of lipids, absorption of minerals, water, and vitamins, and finally describe the regulation of the activity of the small intestine. Before moving on, consider reviewing the organic molecules carbohydrate, protein, and lipids. If you can't list examples of each of these organic molecules, then I would highly suggest that you go back to the chapter on chemistry and do a quick review of these organic molecules. Before we start talking about digestion and absorption in the small intestine, let's review what has occurred up to this point. So food entered the mouth or oral cavity. It was mixed with the saliva. There was mechanical digestion where the teeth, with the help of the tongue, helped break large food particles down into small food particles. Amylase, an enzyme in the saliva, digested amylose, a starch, down to maltose, which is a disaccharide. That is the only chemical digestion that occurs in the mouth, and there is no absorption in the mouth. The bolus, which is formed, was then swallowed and moved into the stomach. In the stomach, pepsin digests protein into shorter polymers called polypeptides. There is no lipid digestion. There is no further carbohydrate digestion, and there is very little absorption of substances by the stomach. Aspirin, alcohol, and a few other substances are absorbed from the stomach, but in terms of nutrients, nothing's really absorbed there. Contents remain in the stomach for quite a while. The peristaltic movements of the stomach help break that bolus that entered the stomach into a heavy cream consistency called chyme, and then that chyme moves into the small intestine. Now that we have reviewed chemical digestion in the oral cavity and the stomach, it is pretty obvious that most chemical digestion is going to occur in the small intestine. And indeed, we find that most carbohydrate and protein digestion occur in the small intestine. Only a minor amount of carbohydrate digestion has occurred in the mouth. Amylase, an enzyme found in saliva, has broken down some of the amylose into maltose. And in the stomach, pepsin has broken down protein or digested protein into shorter chains of amino acids called polypeptides. Lipid digestion, however, occurs exclusively in the small intestine. There was no lipid digestion in the oral cavity, no lipid digestion in the stomach. So all lipid digestion is going to occur in the small intestine. Now, when we talk about digestion, what are we actually talking about? Well, we're talking about chemical reactions that break large molecules down into smaller molecules, which can then be moved into the absorptive cells which line the lumen of the small intestine, and then moved from those absorptive cells into nearby capillaries and lymphatic structures called lacteals. Digestion occurs by the breaking of bonds by water molecules, or what we refer to as hydrolytic reaction. So water is very much involved in digestion. So here we have a large molecule. Water is going to come in and break one of the bonds in that large molecule, releasing two smaller molecules. This is a decomposition reaction involving water, or what we refer to as a hydrolysis reaction or hydrolytic reaction. This is the process by which digestion occurs in the GI tract, regardless of whether we're talking about digestion of carbohydrate or protein or lipid. I would now like to walk you through the process of carbohydrate digestion, and in the subsequent slide, I will talk again about carbohydrate digestion and also absorption. Only single-ring carbohydrates, known as monosaccharides, can be absorbed into the blood. Therefore, enzymes must digest polysaccharides and disaccharides to release monosaccharides for absorption. Polysaccharides ingested in the mouth in the form of a starch called amylose are digested by the enzyme amylase found in the saliva of the mouth to the disaccharide maltose. Any amylose that remains in the chyme as it enters the small intestine is digested to maltose by amylase from the pancreas. Any disaccharides ingested in food pass from the mouth to the stomach and into the small intestine unchanged. Enzymes in the brush border of the absorptive cells digest the disaccharide to monosaccharides, which may now be absorbed into the capillaries of the mucosa. The following figure explains the digestion and absorption of carbohydrate in the small intestine. Any amylose, or digestible starch, that passes into the small intestine is digested to the disaccharide maltose by the enzyme amylase, which is secreted by the pancreas. Any disaccharides consumed in the diet pass from the mouth into the stomach and then into the small intestine unchanged. Brush border enzymes then digest the disaccharides to monosaccharides. Any ingested monosaccharides pass from the mouth, then into the stomach, and then into the small intestine unchanged. Now that the carbohydrate has been digested and is in the form of monosaccharides, the monosaccharides can now be absorbed. Monosaccharides are then transported into the absorptive cell on the lumen side and then transported out of the absorptive cell on the opposite side to enter nearby capillaries, thus completing the process of absorption. This slide outlines the digestion of protein. Amino acids are represented by brown circles. Only single amino acids and small peptide molecules, those containing 2 or 3 amino acids, can pass into absorptive cells. Therefore, enzymes must digest proteins and large polypeptides in order for absorption to occur. Remember that a protein is a long polymer of 50 or more amino acids. Proteins ingested into the mouth pass into the stomach unchanged. In the stomach, pepsin, secreted by chief cells, digests protein to polypeptides. So the pepsin breaks the long polymers of amino acids into shorter ones. The polypeptides then enter the small intestine in chyme, where enzymes from the pancreas called proteases digest the polypeptides, breaking them down into shorter polymers of amino acids, or small polypeptides. Brush border enzymes, provided by the absorptive cells, called peptidases, then digest the small polypeptides into amino acids and some di- and tripeptides, all of which can now enter the absorptive cells. The following figure from your book explains the digestion of protein and the absorption of amino acids and di and tripeptides. As you know, protein passes from the mouth into the stomach unchanged. In the stomach, pepsin digests the protein down into large polypeptides. In some rare cases, those large polypeptides can be transported across the absorptive cells and into the bloodstream. So in rare cases, large polypeptides can actually be absorbed into the blood. Typically, though, pancreatic proteases digest the large polypeptides into di and tripeptides. Those di and tripeptides can be transported into the absorptive cells, some of which are digested by peptidases into amino acids, but prior to moving into the capillaries. Others are absorbed into the capillaries intact. Some di and tripeptides in the lumen encounter peptidases of the brush border, so these are brush border enzymes, which digest them into single amino acids, which are then absorbed into the absorptive cells and then into nearby blood capillaries, completing the absorption of amino acids from the lumen. Now that we have discussed the digestion of carbohydrate and the digestion of protein, let's now turn our attention to the digestion of lipids. If you recall, we did not talk about the digestion of lipids in the mouth or oral cavity. We did not talk about the digestion of lipids in the stomach. And this is because there was none. So lipid passes from the mouth to the stomach and into the small intestine in chyme pretty much unchanged. All lipid digestion occurs in the small intestine. Now, if you recall, lipids do not mix with water, and there's a great deal or great percentage of the chyme is actually water. And so what will tend to happen is the lipid will tend to form big clumps. The function of bile is to break those big clumps into smaller clumps, which exposes a greater surface area for enzymatic breakdown. And we're going to represent an enzyme by the small red arrow. So here we have a big clump of fat that enters the small intestine. The only location where enzymatic breakdown can occur is on the outer surface of that big clump of fat. So in this case, digestion is going to be quite slow because the enzymes can't get to the interior of this fat globule. What bile does is bile breaks the large fat globules down into smaller fat globules, greatly increasing the surface area upon which the enzymes can digest the fat. So the function of bile, produced by the liver and stored in the gallbladder and then secreted into the duodenum when chyme containing fat enters the duodenum, is to emulsify the fat, breaking the large clumps down into smaller clumps, which will increase the rate of lipid digestion. This slide illustrates the digestion of lipid in the small intestine, specifically the digestion of triglycerides, because most of the fat that we consume, most of the lipid that we consume is in the form of triglycerides. Butter, lard, margarine, animal fat, all of those forms of lipid are triglycerides. So triglycerides enter the small intestine in chyme. There they encounter the enzyme lipase, which is secreted by the pancreas. Lipase catalyzes the removal of the first and the third fatty acids from the triglyceride. The products are monoacylglycerol and two free fatty acids. Those molecules then diffuse into the absorptive cells. Once in the absorptive cells, there is quite a complicated process that allows the lipids to pass into the blood of nearby capillaries. This figure from your book explains the process of lipid digestion and absorption. So here we have lipids that have entered the small intestine. We have triglycerides and we have cholesterols, and they form clumps of fat or fat droplets. Bile salts from bile emulsify the large droplets, forming smaller ones. Lipase from the pancreas then digests the triglycerides into two fatty acids and monoglycerides or monoglycerols. You can use the terms interchangeably. So here we have cholesterol, fatty acids, and monoglycerides. The cholesterol uses a carrier protein and enters the absorptive cells. The monoglycerides and the fatty acids can pass into the absorptive cells by simple diffusion. Inside the cells, triglycerides are reformed from the monoglycerides and fatty acids. Then the triglycerides are combined with cholesterol and also proteins to form what are called chylomicrons. The chylomicrons are released from the absorptive cells and they enter not blood capillaries but these lymphatic structures called lacteals, which are shown here in green. So lipids, unlike carbohydrates, unlike amino acids digested and absorbed from protein, bypass the liver and go directly into the blood, because from these lacteals eventually the chylomicrons pass into the subclavian veins. I should add that a small amount of fat does enter the blood capillaries. Very short chain fatty acids escape being packaged into chylomicrons, and they do enter the blood capillaries. But by and large, most fat is packaged into chylomicrons and enters the lacteals. What about other substances that you consume that we have yet to talk about, like the minerals? Well, the minerals pass through the small intestine pretty much intact, and they're absorbed all along the entire length of the small intestine by various processes. Each mineral has different mechanisms by which they are absorbed, which I'm not going to elaborate on. 90% of the water that enters the small intestine is absorbed by osmosis. The remaining 10% passes into the large intestine and helps maintain the fluidity of the contents of the GI tract. Fat-soluble vitamins are absorbed along with the other lipids in the chylomicrons. Fat-soluble vitamins are your vitamins A, D, E, and K. I remember these four by remembering ADEC. What about water-soluble vitamins like your B vitamins and your vitamin C? They're absorbed by simple diffusion. There is one exception, and that would be vitamin B12. Vitamin B12 binds intrinsic factor. If you recall, intrinsic factor is produced by the stomach. Vitamin B12 binds intrinsic factor, and then they enter the absorptive cells by cell-mediated endocytosis. Vitamin B12 cannot enter the absorptive cell by itself. It must bind to intrinsic factor to be transported into the absorptive cells. That is why if the stomach decreases its production of intrinsic factor or ceases to produce intrinsic factor altogether, it will cause a vitamin B12 deficiency in the individual. Let's talk briefly about the regulation of the small intestine. Both the autonomic nervous system and hormones produced by cells that line the small intestine oversee its activity or regulate its activity. The sympathetic nervous system in general decreases the activity of the small intestine, and the parasympathetic nervous system increases the activity of the nervous system through the vagus nerve. Cells of the duodenum, in response to a decrease in pH as chyme enters the small intestine, release the hormone secretin. Secretin passes into the blood, then makes it to the pancreas, where it stimulates the pancreas to release bicarbonate. Bicarbonate to neutralize the acidity of the chyme. So the more chyme that enters the small intestine, the more secretin is released, and the more bicarbonate that is released from the pancreas. If there's a decrease in the amount of chyme that enters the small intestine, there'll be a subsequent decrease in the release of secretin and a decrease in the release of bicarbonate. Another important hormone is cholecystokinin. Cholecystokinin is released by intestinal cells in response to the presence of protein and fat in the small intestine. Cholecystokinin causes the release of bile from the gallbladder. It also causes the release of pancreatic juice. Pancreatic juice contains all of those pancreatic enzymes that digests protein, fat, and carbohydrate in the small intestine. The more fat and protein that enter the small intestine, the more cholecystokinin that is released, and the more bile and pancreatic juice that is released. These hormones therefore ensure that there is complete digestion and absorption of nutrients prior to the passage of the contents of the small intestine into the large intestine. Now let's review the objectives of this screencast. Explain the following processes that occur in the small intestine: digestion and absorption of carbohydrate, digestion and absorption of protein, digestion and absorption of lipids, absorption of minerals, water, and vitamins, and finally describe the regulation of the activity of the small intestine. The topic of the next screencast will be the large intestine.